Hello. In this presentation, I will discuss network perturbation analysis of omics data for complex diseases using convex optimization. This is joint work by Nikos Vlasis and Enrico Glaub from the Luxembourg Center for Systems Biomedicine. Disease-associated molecular perturbations are often localized in biological networks. Finding these network clusters may help us to develop more robust biomarker models. So the question that we addressed in our work is, how can we find clustered gene or protein groups efficiently, accounting both for their predictivity and connectedness in the network? To answer this question, our software called GenePEN uses the following workflow. As input, the user provides a gene or protein expression dataset X, where the rows represent genes and the columns represent the samples. Additionally, class labels I are given to specify the conditions of the samples. For example, patient versus control samples, or samples representing different disease subtypes. Finally, the input contains a table G representing interactions or similarities between the rows in XA, for example, protein-protein interactions. As output, the software will provide a subset of discriminative genes corresponding to rows in A that represent a connected component in graph G that can be interpreted as an altered subnetwork and used as a combinatorial biomarker to predict the class labels for new samples. Regarding the approach behind the software, the idea is to cast the gene selection as an optimization problem, maximizing two quantities: the diagnostic prediction accuracy of the classifier and the connectedness of selected genes in the network. For this purpose, we use a mathematical programming formulation where we minimize a logistic loss function to minimize the prediction error, and additionally minimize a penalty function that enforces the gene grouping in the network. A trade-off parameter lambda, which can be selected using cross-validation, determines the balance between the penalty term and the loss term. The detailed objective function is described on our poster. As an output, the mathematical programming approach will provide an optimized vector of feature weights omega, where components of the vector are set to 0 if the corresponding gene is not selected. If the absolute weight of a component is large, the corresponding gene is relevant for the prediction and well grouped with other selected genes in the network. We compared our approach against previous alternative penalty functions proposed in the literature. These include the ridge penalty, which promotes feature grouping but not a sparse selection of features; the lasso approach, which promotes sparsity but no grouping between features; the elastic net, which is not able to capture local network structures; the grouped lasso approach, which assumes non-overlapping feature groups; the graph kernel approach, where weight signs can introduce biases; and the Oscar approach, where large weights can introduce biases. We first compared the predictive performance of GenePEN and other representative approaches on a test dataset, assessing the average area under the receiver operating characteristic curve in a 5-fold cross-validation for different numbers of selected features. In this evaluation, we obtained similar results for GenePEN compared to the benchmark approaches within one standard deviation from one another. However, the benefits of GenePEN become apparent when we compare the methods in terms of their ability to find grouped features representing locally clustered network alterations. We assess this by comparing the relative size of the largest connected component in the network among the selected features for different total numbers of selected features. Here, GenePEN provides a significantly superior network grouping in all evaluations. Here you can see example results obtained for the different methods when setting the parameters such that approximately 50 genes are selected in total. We can see that only 2 of the 50 genes are grouped together in the network for the lasso approach, 3 for the elastic net and the PEN approach from 2010, 6 for the PEN approach from 2013, but 34 genes for GenePEN. As a practical application, we tested GenePEN on brain transcriptomics data for Parkinson's disease. The network, which you see here on the right, is the largest connected component identified by GenePEN in a protein-protein interaction network. Red nodes in this network highlight overexpressed genes in Parkinson's disease, whereas blue nodes represent underexpressed genes. The color of the node borders from gray to blue represent increasing statistical significance. We can see that in this network, individually significant genes are significantly overrepresented. Furthermore, a literature mining analysis using a pointwise mutual information co-occurrence scoring of the gene names in the network and the term Parkinson's disease in the pub literature database reveals a significant enrichment of positive text mining scores for the selected genes. Overall, the biological results indicate that gene PEN can be helpful to identify sub-network alterations enriched in disease-relevant genes. In summary, our results suggest that integrating prior knowledge from molecular networks into omics data analysis can provide benefits in terms of model robustness and biological interpretability. Our software Gene PEN for this purpose discovers discriminative subnetworks for diagnostic sample classification and enables an interpretation of disease-associated molecular alterations at the network level. On Parkinson's disease transcriptomics data, Gene PEN identifies predictive alterations in subnetworks, which are enriched in individually significant genes and known Parkinson's-associated genes with positive text mining scores.